Hello everyone, and welcome to Chapter 4 of Bio 100. We are going to skip a few chapters and jump right into cells. So happy you could join me for this lecture. Let's go ahead and get started. We have a few objectives for this chapter. The first is to kind of go back and define what a cell is and describe the two general types of cells, to describe the structure and function of cell membranes, to describe several ways in which molecules move across a membrane, to describe how cells are connected and how they communicate with each other, and to describe 9 important landmarks in eukaryotic cells. If you would like, you can find the link to this video in this video's description. It is a nice little recap and not dissimilar to what I'm going to be talking about, except theirs is animated, so it's maybe a little bit more entertaining. So if you want to, you can definitely hop into this link and check that out at your convenience. It's only about a 7-minute long video, so it's pretty good and worth the watch. Alright, so all organisms are made up of cells. You probably know that cells are the smallest unit of life. They are very, very tiny, and most organisms are made up of multiple cells. Some organisms are made up of a single cell, and we'll kind of talk about that here in a little bit. But cells are the smallest unit of life that can function independently and perform all the necessary functions of life, including reproducing itself. Remember from last lecture, we talked about what the characteristics of being a living thing was, and one of those things was that it could reproduce. Another thing about those characteristics is that it must be made up of at least one or more cells. So cells are very important. They make up everything you see, everything you kind of feel in a way. They are everywhere. So something we need to talk about is cell theory. Cell theory is one of the unifying theories in biology. It's a big one, and one that is universally accepted by all biologists. And it states 2 things. The first is that all living organisms are made up of one or more cells. The second is that all cells arise from other pre-existing cells. This means that cells don't just come into an existence out of nowhere. You have to have a parent cell that is going to then split or divide and produce more cells. Sometimes it's 2, sometimes it's 4. I don't think I know of any other situations where it's more or less, but regardless, you have to have a cell coming from another cell. They don't just pop into existence, and that is cell theory. So we have a few types of cells that we need to talk about. Eukaryotic cells and prokaryotic cells. Eukaryotic cells are going to be found in, shockingly, eukaryotes. They have a central control structure called a nucleus. It's kind of not dissimilar to like the brain of the cell, and it's going to contain the cell's DNA. A prokaryotic cell, found in prokaryotes, does not have a nucleus. Its DNA simply resides in the middle of the cell, and we're going to talk about what makes up a eukaryotic cell, what makes up a prokaryotic cell, and where you're going to find these types of cells. So prokaryotes are going to look something kind of like this if we have a nice little illustration. They're going to have these 4 structures. It is what is found in all prokaryotes, and that is a plasma membrane, cytoplasm, which is going to be kind of the fluid on the inside of the cell, DNA floating about in the cytoplasm, and ribosomes. These are going to be little granular bodies in the cytoplasm that convert genetic information into protein structures. So That is very important. And those are the 4 things that all prokaryotes are going to have. Additional things that prokaryotes can have, but not all of them have this, are cell walls, a capsule, pili, which are going to be hair-like projections that help cells attach to other surfaces and sometimes play a role in DNA transfer. And sometimes they'll have a flagellum. A flagellum is going to be kind of like a whip-like projection or a tail that is going to help with that cell in moving. Now, you've probably seen a flagellum before if you've ever seen a sperm cell. So a very classic looking sperm cell has a flagellum on the end. That's that little tail. Eukaryotic cells are going to be what make up pretty much everything you think of that's a living thing. So that includes plants, animals, fungi, mushrooms, and things of that nature. But eukaryotic cells have compartments with specialized functions, almost like organs. And we're going to talk about those, and they help to metabolize things, they help to create energy, they help an organism perform all the functions on a cellular basis that needs to happen. So here we have a lovely little illustration, well, not illustration, this is actually under a microscope, of a eukaryotic versus a prokaryotic cell. So this is an interesting way to view this. If you've never looked at something under a microscope, usually what happens is the subject is dyed with some type of coloring to make it a little bit easier to see. So that is what we're kind of looking at here. So on the left, we have typical eukaryotic cell features, the DNA having a nucleus, or the nucleus surrounding that DNA. We do have cytoplasm, which is going to contain specialized structures called organelles. Now, organelles, as I said before, eukaryotic cells are going to have these structures that act almost like organs, if you want to think of it that way. Not dissimilar to the various systems in a human, these cells have various organs and systems that help them with their cellular functions. Eukaryotic cells are going to be larger than prokaryotic cells, sometimes at least 10 times bigger. Contrary to that, a typical prokaryotic cell, as we said before, will not have a nucleus, and the DNA is in the cytoplasm. So that is something that they do share. They both have a cytoplasm. The internal structure most likely is not organized into compartments like prokaryotic cells, like eukaryotic cells, and they're much smaller than eukaryotic cells. Okay, so here we have an animal cell, which is a eukaryotic cell. We have a few things to go over as far as what is inside of our cell. And we are going to talk about what the functions are of a few of these things. And I believe it's going to be coming up, but we'll see. So we have the nucleus. And inside of the nucleus, that little ball in the very, very center, that is the nucleolus. Now, I don't think you need to know that, but for future knowledge, if you ever wind up on Jeopardy or something, inside the nucleus is the nucleolus, and this is where we're going to have our DNA. Surrounding the entire cell is the plasma membrane. So that's almost like a skin surrounding the cell. Inside of the cell, we're going to have cytoplasm. Cytoplasm is going to be the fluid that is within the cell itself. So it's almost like a jelly-like fluid, but it is a fluid that everything is basically suspended in, all those organelles. We have ribosomes. Those are going to help with protein transfer, essentially. We'll talk about those a little bit later. The one everybody remembers and knows is classically the mitochondria. Remember, that is the, say it with me, powerhouse of the cell that is going to produce ATP. You can just know ATP is energy. Next up, just outside of the nucleus, we're going to have the rough endoplasmic reticulum. Now, the rough endoplasmic reticulum has studded into the walls of it ribosomes, and that is going to be the difference between the rough endoplasmic reticulum and the smooth endoplasmic reticulum, which in this illustration kind of looks like coral, but you'll notice that it does not have ribosomes attached to it. You're going to see a few little kind of like worms, but that is the cytoskeleton that's going to help provide structure to this cell. We have the Golgi apparatus, almost looks like a Wi-Fi symbol. And then we have lysosomes. The lysosomes are going to act in a way like the stomach of the cell. They have digestive enzymes within them, and they are going to be used to digest products that are taken into the cell and also help expel those items. And then the last thing on here, and it does say this structure is not found in plant cells, is a centriole. And I always think the centrioles look like churros or licorice. Just something I've always associated. Now we have our plant cells. So pretty much there's just about everything is the same, aside from just a few things. So structures that are found in plant cells that are not found in animal cells include chloroplast. So make a little note, it is chloroplast, not chloroform. I've had quite a few students write chloroform, and that is not what you find in plant cells. Chloroplast is going to be these little green blobs. If we look at plant cells in lab, you'll see these little blobs kind of moving around. They're very cool to watch, but that is very characteristic of plant cells. Instead of just a plasma membrane, plant cells also have a cell wall. It's a little bit tougher than a plasma membrane, and it's a little bit more durable. We have a vacuole, which occasionally is found in some animal cells, but it is found in plant cells pretty much all the time. So you've got that vacuole right there, and it's very large in the plant cell. So if you could take a little second, read through this. You might want to test yourself. I may post the answers underneath this video if you want to check that. So there's going to be a few of these throughout, so this is just to kind of check your knowledge. When it comes to cells, there's actually quite a few theories. This is one of the other ones. So we have cell theory was the first one, and then we have endosymbiosis theory. Endosymbiosis theory provides the best explanation for the presence of two types of organelles in eukaryotes. So that includes chloroplast and mitochondria. And according to the theory of endosymbiosis, two different types of prokaryotes worked in close partnership. The theory of endosymbiosis is supported by several observations, and here's 4 of them. That chloroplast and mitochondria are similar in size to prokaryotic cells and divide by splitting, or something called fission. Chloroplast and mitochondria have ribosomes. Remember, those guys are closely associated with protein processing. Chloroplast and mitochondria have small amounts of circular DNA. And chloroplast and mitochondrial DNA are more highly related to bacterial DNA than eukaryotic DNA. So this is all just saying that by these observations, it supports this theory of endosymbiosis as to why chloroplasts and mitochondria developed in eukaryotic cells. And so this is specifically saying that mitochondria and chloroplasts together developed in plant and animal cells is what we're talking about here. So we talked about in symbiosis. We need to talk about invagination next. So endosymbiosis is that theory of how mitochondria and chloroplast evolved in eukaryotic cells. So invagination, kind of a similar theory, is also how the origin of other organelles and eukaryotes came about. And it is the idea that the plasma membrane around the cell may have folded in on itself to form the inner compartments, which subsequently became modified and specialized. So that's just another little theory as to how cells, specifically eukaryotic cells, evolved those various organelles inside of themselves. So you can see on the left that nice little illustration of how an ancestral prokaryote kind of pulled in and created a mitochondria. Well, eventually what we now see as a mitochondria into this cell. And then on the right, we see that invagination process, potentially that process where the outside of the cell, the plasma membrane, is folding in and kind of squishing in on itself, and thus is creating the rough endoplasmic reticulum, possibly the nucleus, etc., etc. So it's kind of cool seeing that little potential evolution in cells, which is not something that you can necessarily find in a fossil record, which, you know, we'll get into that maybe eventually. So one of the structures we've already talked about is that plasma membrane, and every cell has that type of border. The function of the plasma membrane is to hold contents of a cell in place. Again, it's not dissimilar to like the skin of the cell. It takes in food and nutrients. So in that way, it also almost acts like the mouth. So it's going to use endocytosis and exocytosis, and we'll talk about those in a little bit, to take in food as well as to expel it. The plasma membrane aids in building and exporting molecules, again, exocytosis, exiting the cell. It allows for interactions with the environment and neighboring cells. And I like this little picture to the right saying that plasma membranes are the gatekeepers of the cell. They allow things in, they also allow things out. But unless you can get past the plasma membrane, you're not coming in. Okay, so let's talk about what allows things in and out, and that is that phospholipid bilayer. So what this means is that it is a phospholipid, and we'll talk about what that means, and is a bilayer. Bi meaning two, so there are two layers. How these layers look is you have these hydrophilic heads, and hydrophilic means that it's attracted to water. So hydro meaning water, philic meaning loving. So it is attracted to water, and that goes on the outermost side of the cell as well as the innermost side of the cell. Remember, there's two layers. So these heads, these hydrophilic heads, are going to be pointing out and inward. Now, attached to the hydrophilic heads is going to be hydrophobic tails. So these tails are going to be facing each other from those two layers. Hydrophobic means water hating or water fearing. This means that it is not attracted to water like those hydrophilic heads, meaning that water is either going to stay in or stay out. It's not going to be able to pass through that layer. So you're going to have the extracellular fluid, which is on the outside of the cell, staying outside, and the intracellular fluid is going to stay on the inside. Here we can see that composition a little bit better. If you're a little bit chemistry minded, maybe this is really exciting for you. I'm not going to go into it very much though. So this is just more for a visualization of those hydrophilic heads and those hydrophobic tails. So molecules embedded within your plasma membrane are going to help perform its functions. This is sort of a way to get around that hydrophobic and hydrophilic heads and tails, so that phospholipid bilayer. So the hydrophobic and hydrophilic forces are going to determine the orientation of proteins in the plasma membrane, and those proteins are then going to help move and transport things into and out of the cell. But this is going to be a very specific process. So several molecules are going to perform important functions in the plasma membrane. We have a few here that I do want you to know. You have carbohydrate chains, which are going to provide a fingerprint for identification by other cells. You have cholesterol, which is embedded in that phospholipid bilayer that helps the membrane retain its flexibility. You have receptor proteins, and you'll notice that's nice and embedded, and you have an entrance and an exit going through that phospholipid bilayer that is going to allow a passage, if you will, through that gate, for lack of a better term, going back to what we said before. So these receptor proteins are going to bind to external chemicals that regulate processes within the cell. We have recognition proteins, which are going to provide a fingerprint for identification by other cells, and transport proteins, which are going to provide a passageway for molecules through the cell. So this is going to act as quite literally a gate or a checkpoint where you can go in and out. But there are only going to be particular things that are allowed in and out. And then we have membrane proteins, which are going to accelerate intracellular and extracellular reactions on the plasma membrane. If your plasma membrane is not acting the way that it should, it can cause diseases. And one of those diseases is cystic fibrosis. Cystic fibrosis, or CF, is where your body produces a thick and sticky mucus and it collects in the lungs. This is going to cause an impairment of lung function and increase the risk of bacterial infection. The reason that that happens is because with that thick mucus being stuck in the lungs and in the throat and the trachea, so essentially just the whole respiratory system, with that thick, sticky mucus just sitting in there, if you breathe in bacteria, it has the best place to hang out and reproduce like crazy. So getting sick is a very big deal when you have CF. So some ways that you can kind of soothe or treat cystic fibrosis. The young lady on the left is getting a thumping on her chest or back to loosen the mucus up and then be coughed out, expelled, or swallowed. The young lady on the right has a vest that acts by inflating and deflating rapidly and can have a similar effect in the course of about a 20-minute session instead of getting your back or your chest thumped on. When it comes to cystic fibrosis, that is a condition where the plasma membrane is having a problem transporting chloride ions, and that is caused from a genetic condition. Contrary to that type of thing, pharmaceutical drugs can also alter your membrane function. An example that we're showing you here is beta blockers. Beta blockers, in stressful situations, the adrenal glands will pump out adrenaline, and then the adrenaline, which is a hormone that is essentially your fight or flight chemical. It's going to get you all hot and sweaty and ready to go, ready to fight or ready to run away, cannot enter the cell when beta blocking medications bind to the cell's beta receptors. So it is going to inhibit the adrenaline from happening, I guess, or from binding is probably a better term here, which means it's going to reduce anxiety symptoms. So if you are someone who is easily anxious or has panic attacks or suffers from anxiety, then beta blockers are going to help you manage that anxiety. So this is going to be something that's a little bit more than you have to go talk up in front of a class, you know, in the little rush of adrenaline before you have to present something to a group of people. This is something that happens on a daily basis potentially. That is not a fun time. It is not something you want to experience, but beta blockers will help by blocking those binding sites. So plasma membrane surfaces, as I mentioned before, have something like a fingerprint that is going to identify the cells to each other, in essence. This is a way that your body helps fight off infections, such as bacteria or viruses, sometimes parasites. What happens is your cells are able to identify through that fingerprint who is supposed to be there and who isn't. And if it's not supposed to be there, it's going to cause an immune response, and your body is going to start fighting off whatever that foreign pathogen is. In the case of the AIDS-causing virus HIV, it uses the molecular fingerprints on plasma membranes to infect an individual's cells. These same molecule markers are also the reason that it's extremely unlikely that you can catch HIV infection from casual contact, like hugging, shaking hands, or sharing a drinking glass. The specific molecule markers involved in infection by HIV belong to a group of identifying markers called clusters of differentiation. It's abbreviated as CD markers, which you can see that on this little diagram or this little illustration. These markers, marker molecules, are proteins embedded in the plasma membrane that enable the cell to bind to outside molecules and sometimes transport them into the cell. One CD marker, called the CD4 marker, is found only on cells deep within the body and in the bloodstream, such as immune system cells and some nerve cells. It is the CD4 marker in conjunction with another receptor that is targeted by HIV. If the virus can find a cell with a CD4 marker, it can infect the cell, and because the CD4 marker never occur on the surface of the skin, casual contact, such as touching, is extremely unlikely to transmit the virus. Even if millions of HIV particles are present on a person's hands, they just can't gain access into one of the other person's surface cells. So that is why HIV cannot be transmitted through casual contact. But it can be contracted through fluids such as blood, vaginal fluids, or semen. So moving on to how things move into and out of cells, the first thing we're going to talk about is passive transport. It is the spontaneous diffusion, which when you see the word diffusion, I want you to immediately think movement. Diffusion is just the movement of molecules across a membrane. And passive transport, as the name implies, is going to be passive. There is no energy that is pushing those molecules to move. There are two types of passive transport. The first is simple diffusion, and the second is facilitated diffusion. For simple diffusion, it's going to be where molecules pass directly through the plasma membrane without the assistance of another molecule. And facilitated diffusion is where molecules move across the plasma membrane with the help of a channel or a carrier molecule. The next type of movement is going to be osmosis. This is going to be the passive diffusion of, and make sure you know this, water. So specifically osmosis is going to have to do with water across a membrane. And there are a few different types of movement. You have isotonic, where water movement is going to be balanced. The amount of water moving into a cell is the same as moving out of the cell. You have a hypotonic solution, where water is going to diffuse into the cell until the cell bursts or swells. In the case of our animal cell, over on the right there, that illustration, we have a red blood cell versus a plant cell. And so that red blood cells from an animal, obviously, and our plant cells from a plant. With hypotonic solutions, water is going to enter that plant cell and it's just going to swell. In the animal cell, the red blood cell, a hypotonic solution is going to cause the water to make the cell swell and eventually burst. So kind of pop like a overripe balloon, like an overripe water balloon. In the hypertonic solution, so make sure you pay attention to how both of those are spelled. Hypo has an O and hyper has an E. This is going to be water is diffusing out of the cell and our cell is going to shrink and shrivel. So cells in solution. So what we were just talking about is tonicity. That is the relationship between the concentration of solutes inside the cell and solutes outside of the cell in a solution. And essentially, what we were just talking about is exactly what's on this slide. It's just phrased in more of a definition. So isotonic versus hypo and hypertonic. Exactly what we were just talking about. So here we have an example of osmosis in action. We are using a dehydrated celery stalk. So he is thirsty for some water. You can see inside where the cells have the dissolved molecules and there is water, but our dehydrated celery contains many dissolved molecules or solutes relative to the water molecule. So there's more solutes than there is water, which is going to cause our celery stalk to kind of wilt and become limp and not how celery should be or how you want it to be. So when we place our celery stalk into some distilled water, you're going to notice he perks up, and that is because the distilled water contains fewer dissolved molecules than the celery cells. So that water is going to move into our celery stalk. The water molecules diffuse into the celery, so they're moving into the celery and equalizing the water concentration inside and outside of the cells. So we have this nice isotonic situation happening. That's very good. We want that. If, however, we place the celery into salt water, the salt water is going to contain more dissolved molecules than the celery cells. Water molecules are going to diffuse out of the celery in that instance. The celery is going to become more shriveled. Now, what I want you to think of when you think of this process is that we are moving, and I would write this down, highlight it, whatever you need to do. We are moving from an area of high concentration of something to an area of low concentration. So by having fewer molecules, dissolved molecules, inside of the celery compared to outside of the cell, we are going to have water move out of the cell because it's going to go to that other area of low concentration of water. And you might even have some salt molecules move into the celery, but more importantly, water is going to be moving out and thus causing our celery to become more shriveled. In that other example from just before, where our celery was being placed in distilled water, we had water moving in because there was less water inside of the celery And so that water was moving from the outside to the inside to kind of compensate for that until there was a nice isotonic, equalized situation happening. So in active transport, cells use energy to move small molecules into and out of the cell. Active transport is sort of the opposite of passive transport, where it requires energy to move something. And in this case, we are using ATP. So ATP is, and forever will be, energy. So active transport occurs when the movement of molecules into and out of a cell requires the input of energy. In secondary active transport, the transport proteins moves one molecule against its concentration gradient. So that's something else. So that concentration gradient, typically we're going to move down it. If you're moving up the concentration gradient, you're kind of moving against the grain. Moving down the concentration gradient is going to be going from an area of high concentration to an area of low concentration, and vice versa. For moving against the concentration gradient, you're going from low to high. So that is going to require energy to do that. This is going to be powered by the simultaneous flow of another molecule down its concentration gradient. So it's going to kind of piggyback on that natural way of concentration gradients. So there are two distinct types of active transport, primary and secondary. Primary active transport is going to use ATP directly, while secondary active transport is going to transport proteins obtained energy in an indirect method. So essentially it's just not directly using energy in the form of ATP. Okay, I said we'd talk about this, and this is endocytosis and exocytosis. These are going to be used for the bulk transportation of particles into and out of a cell. Endocytosis, you can think of it as into the cell. Whenever you see the word cyte, that just means cell. Endo just means in. So we're just talking about going into the cell, the process of going into the cell. It is the absorption of large particles into a cell. This is going to be through engulfing large particles with the plasma membrane. Remember, I said the plasma membrane is kind of like the mouth and skin of a cell. There are three types of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. And then we have exocytosis, which is the exiting of something out of the cell. So this is the exportation or exit of large particles out of a cell. So talking a little bit about phagocytosis and pinocytosis, you have phagocytosis on the left, where you can see that cell essentially just engulfing it. Eventually that cell is going to wrap itself around whatever that item is and engulf it entirely. It's kind of cool to watch, and I might try to find a video for you guys eventually. And then on the right, you can see that process of phagocytosis a little bit more as an illustration. So you've got that particle, and you can see that plasma membrane starting to open up and then wrap around that item. So it creates kind of like a pocket. From there, it creates, in a way, a vesicle that can then be digested by like a lysosome or something like that. So it creates almost like a little food pouch, if you will, to then be processed. I realize on that last slide I didn't state what pinocytosis is. It's essentially the same thing as phagocytosis. It's just that phagocytosis is going to engulf solids, while pinocytosis is going to engulf liquids. So moving into receptor-mediated endocytosis. So again, we're still bringing something into the cell, but this is receptor-mediated. This is a type of endocytosis by which cells engulf specific particles. An example of this is that it's a process that's important in the ability of your liver to remove cholesterol from your bloodstream. It does show this little process, however, I don't need you to know the process. Just know what it is and kind of why it happens. So remember how endocytosis has the plasma membrane wrapping around whatever molecule or whatever it is that is coming into the cell. Exocytosis still has that plasma membrane bit wrapped around that little package there. So with exocytosis, it's going to be pushing that vesicle, that package with the molecules inside of it, to the very edge of the cell where that plasma membrane that's wrapped around that package is going to fuse with the actual plasma membrane surrounding the cell, and thus have whatever is inside exit the cell after that plasma membrane connects with the other plasma membrane. Moving on to the nucleus. So that was everything plasma membrane that we could possibly get into. Let's move into the nucleus. The nucleus is the cell's genetic control center. Remember, I said it's not dissimilar to the brain of the cell. It's very, very important. It's almost like the brain and the heart in a way. The nucleus is the largest and most prominent organelle in most eukaryotic cells. It's going to be what you typically are able to see if you look at a cell under a microscope. The nucleus has two primary functions. It is genetic control and the storehouse for hereditary information. So that just means your DNA. The structure of the nucleus has three parts. So the nuclear membrane, which is two bilayers that also contain pores, so ways for things to enter and exit. It's going to also contain chromatin, which chromatin are thin fibers of DNA which carry all hereditary information. And then the other thing that it's going to contain is the nucleolus. I mentioned the nucleolus before. It is what is on the very inside of the nucleus. And this is the area of the nucleus where ribosomal subunits are assembled. So again, back to those ribosomes. Moving on to the cytoplasm and the cytoskeleton, which both form the cell's internal environment. They are both going to provide support and can generate motion in and out of the cell. So the functions of the cytoskeleton include acting as the inner scaffolding of the cell, so not dissimilar to how our skeleton and bones act as leverage and basically a structure to hold everything up inside us. It also provides shape and support. It controls intracellular traffic flow and enables movement. It is going to come in the form of three types. You've got microtubules, which are going to be hollow tubes. They're a little bit bigger and thicker. Intermediate filaments, which are going to be durable and rope-like, and microfilaments, which are going to be the smallest of the three, but they are going to be long, solid, rod-like fibers that are going to help with cell contraction and cell division. And we'll talk about cell division a little bit later in the semester. Next, we have cilia and flagella. These are both going to be used for movement of the cell itself. Cilia are going to be short projections that beat swiftly to move fluid along and past a cell. So it's sort of moving what is outside of the cell along the cell's surface. Flagella, which we talked about before, are long microtubule-based structures that move cells through their environment. So they're like a little whip-like tail that's going to propel the cell through its environment. On to mitochondria. Again, it is the powerhouse of the cell. It is where the cell's energy is converted into something that can be used, like ATP. The function is to act as an all-purpose energy converter and to harvest energy to be used for cellular functions. There are different parts of the mitochondria, however, I do not need you to know them. Something interesting about mitochondria is that mitochondria has its own special DNA, which has been a really cool thing for scientists to discover, that mitochondria has mitochondrial DNA specific to itself. The number of mitochondria in your cells are going to vary. You are going to have substantially more mitochondria found in your muscle cells than, say, in your skin cells. That is because your muscle cells need substantially more mitochondria. So the number of mitochondria per cell is going to vary with your types of cells. And even though I said your muscle cells are going to have a lot, the winner of the type of tissue that has the most mitochondria is going to be your liver. So liver cells actually contain more mitochondria than even your muscle cells. As we learned in our earlier discussion of endosymbiosis, mitochondria may very well have existed billions of years ago as separate single-celled bacteria-like organisms. They are similar to bacteria in size and shape and may have originated when symbiotic bacteria took up permanent residence within other cells like those early eukaryotic cells. Perhaps the strongest evidence for this is that mitochondria have their own DNA, as I mentioned on that last slide. Anywhere from 2 to 10 copies of its own little ring-shaped DNA are mixed in among the approximately 3,000 proteins in each mitochondrion. This DNA carries the instructions for making 13 important mitochondrial proteins necessary for metabolism and energy production. So we're always taught that our mothers and fathers contribute equally to our genetic composition, but actually your mom contributes just a little bit more, and that is due to the fact that the mitochondria that you have is the same as your mom's. You do not inherit your father's mitochondrial DNA. You always inherit your mother's. So it doesn't matter if you're a boy or a girl, you always get your mom's mitochondria. Moving on to my personal favorite of all the organelles inside of cells, and that is lysosomes, because they're essentially just the garbage disposals or stomachs of the cell. So their function is to act as a floating garbage disposal for cells, digesting and recycling cellular waste products and consumed material. This is because they have a digestive enzyme and acid inside of them, which is just super cool. They're just walking acid bombs. In the endoplasmic reticulum, or the ER, cells will build proteins and disarm toxins. Their functions are to produce and modify molecules to be exported to other parts of the organism, and to break down toxic chemicals and cellular byproducts. And we do have 2 types, the rough ER and the smooth ER. The rough ER, or rough endoplasmic reticulum, has the function of modifying proteins that will be shipped to other locations in the endomembrane system, the cell surface, or outside of the cell. And again, when you think of the difference between the rough ER and the smooth ER, the rough ER is going to be covered in ribosomes. And remember, ribosomes have everything to do with protein making and protein packaging and protein transportation and all that good stuff. The smooth ER, in comparison, has the function of synthesizing lipids, or you can think of those as fats, such as fatty acids, phospholipids, and steroids. It also serves the purpose of detoxifying molecules such as alcohol, drugs, and metabolic waste products. And it is called smooth ER because it does not contain any ribosomes on its surface. Next up is the Golgi apparatus. This is where we process products for delivery throughout the body. Its function is to process and package proteins, lipids or fats, and other molecules for export to other locations inside or outside of the cell. I always like thinking of the Golgi apparatus as kind of being like a mailroom. It's sort of deciding where things go, it's packaging them up, and specifically it's going to package it up into a transport vesicle that you can see there that is illustrated. All right, so here we have an illustration of the endomembrane system, which we can separate out into about 5 steps. What the endomembrane system is the producing, packaging, and transporting of molecules. So that first step that you see happening at the rough ER, we have the transport vesicle budding off of the smooth or rough ER. So either one, but the one that I can kind of see a little bit better is the rough ER, but it happens at both. The second step is where the transport vesicle that just popped off of the smooth or rough ER fuses with the Golgi apparatus and then dumps the contents inside of it. The third step is the Golgi apparatus modifying the molecules as they move through its successive chambers. The fourth step is where the modified molecules, after going through the Golgi apparatus, buds off from the Golgi apparatus as a second transport vesicle. And then the fifth step is where the vesicle may fuse with the plasma membrane of the cell, and thus dumping the contents outside of the cell for delivery elsewhere in the organism. So it is processed inside of the cell and then gets dumped out of the cell to be used elsewhere. So moving back to plant cells, so what we were looking at before was processes that can happen in eukaryotic cells, but we were visualizing it more in an animal cell. So moving back into plant cells, the cell wall is going to provide additional protection and support for our plant cells, very heavily on the support. It's going to give plant cells a very rigid look. They almost look octagonal and like they have points on the edges rather than an animal cell, which is going to be more round in appearance. So then we have this term here called plasmodesmata. Try saying that 5 times fast. These are going to be tube-like channels that connect plant cells to each other. While animal cells do connect with each other in some capacity, they're not quite like plant cells in this instance where they have a little bit more of a cohesion to them through these plasmodesmata. On to vacuoles. They are multi-purpose storage sacs for plant cells. There are 5 important roles of the central vacuole. The first is nutrient storage. The vacuole is going to store hundreds of dissolved substances, including amino acids, sugars, and ions. The second function is waste management. The vacuole is going to retain waste products and degrade them with digestive enzymes, not dissimilar to the lysosome. The third is predator deterrence. The poisonous, nasty tasting materials that accumulate inside the vacuoles of some plants make a powerful deterrent to animals that might try to eat parts of the plant. So it might end up tasting bitter or sour, just not good in general. That's kind of how you can tell if something's poisonous as well. Sexual reproduction is the next function. And that is the vacuole may contain pigments that give some flowers their red, blue, purple, or other colors, enabling them to attract birds and insects that help the plant reproduce by transferring pollen. And the fifth function is physical support. So high concentrations of dissolved substances in the vacuole can cause water to rush into the cells through the process of osmosis. Remember, that is the movement of water. The increased fluid pressure inside the vacuole can cause the cell to enlarge a bit and push out the cell wall. This process is responsible for the pressure, which in plants is called turgor pressure, that allows stems, flowers, and other plant parts to stand upright. So if you notice that, the flowers that you got your mom for Mother's Day or birthday or, to say sorry or something are wilting, it's because that turgor pressure is no longer in that stem. And that is what causes flowers to stand upright. Okay, on to chloroplasts, little green buddies in there that are super adorable to see under a microscope. Chloroplasts are the plant cell's solar power plant, which, again, try saying that 5 times fast. Chloroplasts resemble photosynthetic bacteria and are possessing a dual membrane and circular DNA. Their function is to be the site of photosynthesis, which is the conversion of light energy into chemical energy, specifically into sugars. Okay, and here we have a review of cell structures, what they do, their function, as well as what type of cell you're going to find it in as far as eukaryotic cells, so animals versus plants. We've got our animal cell here, all labeled out. And if you'd like to, you can continue looking at these slides. I will pause on each one, but I won't talk over them, just so you can kind of look at them and see if you want to test your knowledge with a few of these questions. Otherwise, if you would like to end this lecture now, you're more than welcome to. Thank you for joining me for Chapter 4. I hope you enjoyed it. Cells are kind of interesting, hard to conceptualize, but very, very important to understand. So I will talk at you next time in the next chapter. Make good choices.